In today's lecture, we will discuss about Golgi bodies, structure and function, vesicular transport and types of transport vesicles, mechanism of vesicular transport, structure and function of ER and lysosomes, and in the end about the comparative features of endoplasmic reticulum, Golgi apparatus and lysosomes. At the outset, we talk about Golgi apparatus. The Golgi complex, Golgi body or simply the Golgi is an organelle found in most eukaryotic cells. They were identified in 1897 by Italian physician Camillo Golgi and named after him in 1898. The Golgi apparatus resides at the intersection of the secretory, lysosomal and endocytic pathways. It is of particular importance in processing proteins for secretion. Now about the structure of Golgi apparatus. Golgi apparatus is made up of a series of compartments consisting of two main networks, the cis-Golgi network and the trans-Golgi network. The cis-Golgi network is a collection of fused, flattened, membrane-enclosed discs known as cisternae, originating from vesicular clusters that bud off the endoplasmic reticulum. A mammalian cell typically contains 40 to 100 stacks. This collection of cisternae is broken down into cis, medial and trans compartments. The trans-Golgi network is the final cisternal structure from which proteins are packaged into vesicles destined to lysosomes, secretory vesicles or the cell surface. The trans-Golgi network may act as an early endosome in yeast and plants. There are structural and organizational differences in the Golgi apparatus among eukaryotes. Among eukaryotes, the subcellular localization of the Golgi apparatus differs. In mammals, a single Golgi apparatus complex is usually located near the cell nucleus close to the centrosome. In yeast, multiple Golgi apparatuses are scattered throughout the cytoplasm, as observed in Saccharomyces cerevisiae. In plants, Golgi stacks are not concentrated at the centrosomal region and do not form Golgi ribbons. Common feature among Golgi is that they are adjacent to ER exit sites. The Golgi apparatus tends to be larger and more numerous in cells that synthesize and secrete large amount of substances. For example, the antibody secreting plasma B cells of the immune system. In all eukaryotes, each cisternal stack has a cis entry phase and a trans exit phase. The compartmentalization of the Golgi apparatus is advantageous for separating enzymes, thereby maintaining conjugative and selective processing steps for protein formation. Now the functions of Golgi apparatus. The Golgi apparatus is a major collection and dispatch station of protein products received from the ER. The Golgi can be thought of as similar to a post office. It packages and labels items which it then sends to different parts of the cell or to the extracellular space. They are involved in lipid transport and lysosome formation. Individual stacks have different assortment of enzymes allowing for progressive processing of cargo proteins as they travel from the cis to the trans-Golgi phase. Enzymatic reactions within the Golgi stacks occur exclusively near its membrane surface, where enzymes are anchored. An important function of Golgi apparatus is in the formation of proteoglycans. After Golgi apparatus, we will briefly talk about structure and function of endoplasmic reticulum. Endoplasmic reticulum, which is an interconnected network of membrane vesicles or small containers used by cells to transport or hold materials. The surface of the ER is basically a workspace for the cell. To maximize the amount of surface area that is available, the membranes are folded tightly rather than being one flat surface. The endoplasmic reticulum is composed of sac-like structures called cisternae that are folded and bound together by a cytoskeleton. There are two types of endoplasmic reticulum: smooth and rough endoplasmic reticulum. Smooth ER gets its name from its smooth folded surface, while as rough endoplasmic reticulum gets its name because its surface is studded with ribosomes, making it appear bumpy. Smooth and rough ER are often attached to each other but can be distinguished by the shape. Smooth ER is tubular while rough ER is flatter. Now we will talk about the smooth ER. Smooth ER consists of a long network of a folded tube-like structure. The interior of smooth ER is called the lumen, which is enclosed by a phospholipid membrane just like the membrane that encloses the entire cell. The number of smooth ER units in a cell depends on the type of cell and what its manufacturing needs are. Smooth ER also makes or distributes products according to its location. In muscle cells, it releases calcium ions, while in cells of endocrine system, smooth ER makes steroid hormones. The main function of the smooth ER is to make cellular products like hormones and lipids, distributes those products throughout the cell and to places in the organism, regulates and releases calcium ions and processes toxins. The smooth ER is an organelle found in both animal cells and plant cells. Now the rough endoplasmic reticulum. The surface of the rough ER, also called ergastoplasm, is studded with protein manufacturing ribosomes, giving it a rough appearance. The binding site of the ribosome on the rough ER is the translocon. However, the ribosomes bound to it at any one time are not a stable part of this organelle structure, as they are constantly being bound and released from the membrane. A ribosome only binds to the rough ER once a specific protein nucleic acid complex forms in the cytosol. This special complex forms when a free ribosome begins translating the messenger RNA of a protein destined for a secretory pathway. The protein is processed in the ER lumen by an enzyme called a signal peptidase, which removes the signal peptide. Ribosomes at this point may be released back into the cytosol. However, non-translating ribosomes are also known to stay associated with translocons. The membrane of the rough ER forms large double membrane sheets that are located near and continuous with the outer layer of the nuclear envelope. Although there is no continuous membrane between the endoplasmic reticulum and the Golgi apparatus, membrane-bound vesicles shuttle proteins between these two compartments. Vesicles are surrounded by coating proteins called COP1 and COP2. COP2 targets vesicles to the Golgi apparatus and COP1 marks them to brought back to the rough ER. The rough ER works in the concert with the Golgi complex to target new proteins to their proper destination. Now we will talk about the functions of rough endoplasmic reticulum. The main functions are manufacturing of lysosomal enzymes, secreted proteins and integral membrane proteins, initial glycosylation and normal linked glycosylation. After Golgi apparatus and endoplasmic reticulum, now we will learn about the vesicular transport. The vesicles that leave the rough air are transported to the cis phase of the Golgi apparatus, where they fuse with the Golgi membrane and empty their contents into the lumen. Once inside the lumen, the molecules are modified and then sorted for transport to their next destinations. Those proteins destined for areas of the cell other than either the endoplasmic reticulum or the Golgi apparatus are moved through the Golgi cisternae towards the transphase to a complex network of membranes and associated vesicles known as the trans-Golgi network. This area of the Golgi is the point at which the proteins are sorted and shipped to their intended destination by their placement into one of at least 3 different types of vesicles, depending upon the signal sequence they carry: the exocytotic vesicles, secretory vesicles, and number 3, lysosomal vesicles. Vesicles are similarly involved in the transport of materials taken up at the cell surface. Vesicular transport is thus a major cellular activity, responsible for molecular traffic between a variety of specific membrane-enclosed compartments. The selectivity of such transport is therefore key to maintaining the functional organization of the cell. For example, lysosomal enzymes must be transported specifically from the Golgi apparatus to lysosomes, not to the plasma membrane or to the air. The proteins are transported within vesicles, so the specificity of transport is based on the selective packaging of the intended cargo into vesicles that recognize and fuse only with the appropriate target membrane. The first step in vesicular transport is the formation of a vesicle by budding from the membrane. The cytoplasmic surfaces of the transport vesicles are coated with proteins, and it appears to be assembly of these protein coats that drives vesicle budding by distorting membrane conformation. 3 kinds of coated vesicles which appear to function in different types of vesicular transport have been characterized. The first to be described were the clathrin-coated vesicles, which are responsible for the uptake of extracellular molecules from the plasma membrane by endocytosis, as well as the transport of molecules from the trans-Golgi network to lysosomes. 2 other types of coated vesicles have been identified as budding from the ER and Golgi complex. These vesicles are called non-clathrin-coated or COP-coated vesicles. COP stands for the coat protein. One class of these vesicles, COP second coated vesicles, bud from the ER and carry their cargo forward along the secretory pathway to the Golgi apparatus. The coats of clathrin-coated vesicles are composed of 2 types of protein complex, clathrin and adapter proteins, which assemble on the cytosolic side of membranes. The binding of clathrin to membranes is mediated by a second class of proteins called adapter proteins. Incorporation of lysosomal proteins into clathrin-coated vesicles. Proteins targeted for lysosomes are marked by mannose-6-phosphatases, which bind to mannose-6-phosphate receptors in the trans-Golgi network. The mannose-6-phosphate receptors span the Golgi. Now the vesicle fusion. The fusion of a transport vesicle with its target involves 2 types of events. First, the transport vesicle must specifically recognize the correct target membrane. For example, a vesicle carrying lysosomal enzymes has to deliver its cargo only to lysosomes. Second, the vesicle and target membrane must fuse, thereby delivering the contents of the vesicle to the target organelle. Analysis of protein involved in vesicle fusion is described by the general model called the SNARE hypothesis, in which vesicle fusion is mediated by interactions between specific pairs of proteins called SNARE on the vesicle and target membrane V SNAREs and T SNAREs respectively. we will talk about lysosomes. Lysosomes are membrane enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, proteins, nucleic acids, carbohydrates and lipids. Lysosomes function as the digestive system of the cell, serving both to degrade material taken up from outside the cell and to digest absolute components of the cell itself. Discovered and named by Belgian biologist Christian de Dieu in 1974. Lysosomes thus represent morphologically diverse organelles defined by the common function of degrading intracellular material. Enzymes of the lysosomes are synthesized in the rough air. The enzymes are released from the Golgi apparatus in small vesicles, which ultimately fuse with acidic vesicles called endosomes, thus becoming full lysosomes. Lysosomes are interlinked with three intracellular processes, namely phagocytosis, endocytosis and autophagy. Lysosomes are cellular organelles that contain acid hydrolase enzymes that break down waste materials and cellular debris. Described as the stomach of the cell, lysosomes digest excess or worn out organelles, food particles and engulfed viruses or bacteria. The membrane around a lysosome allows the digestive enzymes to work at the pH they require. Lysosomes fuse with the autophagic vacuoles, phagosomes, and dispense their enzymes into autophagic vacuoles, digesting their contents. They are frequently nicknamed suicide bags or suicide sacs. The size of lysosomes varies from 0.1 to 1.2 micrometer. At pH 4.8, the interior of the lysosomes is acidic compared to the slightly basic cytosol. The lysosome maintains this pH differential by pumping in protons, hydrogen ions from the cytosol across the membrane via protein pumps and chloride ion channels. The lysosomal membrane protects the cytosol and therefore the rest of the cell from the degradative enzymes within the lysosome. The cell is additionally protected from any lysosomal acid hydrolases that drain into the cytosol, as these enzymes are pH sensitive and do not function well or at all in the alkaline environment of the cytosol. This ensures that cytosolic molecules and organelles are not destroyed in case there is leakage of the hydrolytic enzymes from the lysosomes. Now, the formation of lysosomes. Many components of animal cells are recycled by transferring them inside or embedded in sections of membrane. For instance, in endocytosis, more specifically macropinocytosis, a portion of the cell's plasma membrane pinches off to form a vesicle that will eventually fuse with an organelle within the cell. The production of lysosomal proteins suggests one method of lysosome sustainment. Upon exiting the air and entering the Golgi apparatus via vesicular transport, a specific lysosomal tag, mannose-6-phosphate, is added to the peptides. The presence of these tags allow for binding to mannose-6-phosphate receptors in the Golgi apparatus, a phenomenon that is crucial for proper packaging into vesicles destined for the lysosomal system. Now, the lysosomal acid hydrolases. Lysosomes contain about 50 different degradative enzymes that can hydrolyze proteins, DNA, RNA, polysaccharides and lipids. Mutations in the genes that encode these enzymes are responsible for more than 30 different human genetic diseases, which are called lysosomal storage disease. For example, Gaucher's disease. All of the lysosomal enzymes are acid hydrolases, which are active at the acidic pH about 5 that is maintained within lysosomes. Now, the endocytosis and lysosome formation. One of the major functions of lysosome is the digestion of material taken up from the outside the cell by endocytosis. Lysosomes are formed by the fusion of transport vesicles budded from the trans-Golgi network with endosomes, which contain molecules taken up by endocytosis at the plasma membrane. Material from outside the cell is taken up in clathrin-coated endocytic vesicles, which bud from the plasma membrane and then fuse with early endosomes. Membrane components are then recycled to the plasma membrane, and the early endosomes gradually mature into late endosomes, which are the precursors to lysosomes. Now, the phagocytosis and autophagy. In addition to degrading molecules taken up by endocytosis, lysosomes digest material derived from two other routes: phagocytosis and autophagy. In phagocytosis, specialized cells such as macrophages taken up and degrade large particles, including bacteria, cell debris and aged cells that need to be eliminated from the body. Such large particles are taken up in phagocytic vacuoles, phagosomes, which then fuse with lysosomes, resulting in digestion of their contents. Autophagy. The gradual turnover of the cell's own components. The first step of the autophagy appears to be the enclosure of an organelle, example a mitochondrion, in a membrane derived from the ER. The resulting vesicle, an autophagosome, then fuses with a lysosome and its contents are digested. Now, the pathogenesis. Lysosomes are responsible for a group of genetically inherited disorders called lysosomal storage disease. They are a type of inborn errors of a metabolism caused by malfunction of one of the enzymes. And in the end, about the comparative features of endoplasmic reticulum, Golgi apparatus and lysosomes. Cells have extensive sets of intracellular membranes which together compose the endomembrane system. The endomembrane system or intracellular membranes are separate compartments with specific functions. These structures do exchange membrane material, however, via special type of transport. Vesicles also allow the exchange of membrane components with a cell's plasma membrane. With this, we conclude today's lecture on Golgi apparatus, ER, vesicular transport and lysosomes. Hope you enjoyed and thank you.